Thank you so much for having me. My name is Kimberly Goodspeed. I'm a child neurologist and neurodevelopmental disability specialist at UT Southwestern in Dallas, Texas. My clinical practice is at Children's Health Dallas in the Center for Autism and Developmental Disabilities. I'm very grateful and very excited that Simon Searchlight invited me to come speak at the symposium today to talk about a topic that's really near and dear to my heart, about preparing for clinical trials in neurodevelopmental disabilities. And so we'll go on a little bit of a journey today, but the biggest take-home message that I really want you to leave here with is that we're going to begin with the end in mind. So we always want to have that final goal and that, end goal in mind as we're preparing and moving along through any kind of research or clinical program. And so to get there, there's a number of things that I think we can talk through of ways to set ourselves up for success. So we're going to start today by just going through some basics of kind of the lay of the land with developmental disabilities as well as autism, and then go through and touch on precision therapies just for a moment, discuss a little bit more in depth on clinical trials, different types of trial designs, and then finally ending with where we'll really end up starting any clinical program, which is with our observational studies. So to begin today, I just want to lay the land and say, prevalence of developmental disabilities is very high. 1 in 6 children in the US between the ages of 3 years and 17 years have some degree of a developmental disability. And now 1 in 54 kids is diagnosed with autism spectrum disorder, which has just been massively on the rise over the last decade or so. And if we put that in the context of your average school classroom, so in Texas, our special education classrooms are around, 8 kids or so. And our general education classrooms usually have 20 to 25 children in them. And so if you think about that general education classroom, it's really every other classroom in the US that has a child with autism. And then also, if we think about our developmental disabilities, that's multiple children within each classroom that have some degree of a disability. So it's a big problem. It's affecting tons of families and individuals. And it's a problem that we'd all like to tackle with a lot of different options. As we're moving through, over time, research has really found these strong genetic links between certain genes that affect brain function or development and our developmental disabilities. There are certain genes that are very prevalent and commonly mutated or have variants within them that cause dysfunction of that protein within the realm of neurodevelopmental disabilities as well as autism. What we've learned from that is that there's over 100 that are specifically linked with autism. And many of these genes are associated with either brain cell development or maturation, so where the brain cells get to and how they function, how they do their jobs, or how they talk to each other, so maybe through receptors or different types of channels that help brain cells talk to each other, or by regulating how other brain cells are expressing different genes or turning on different genes at different times, at different time points in development. And so the landscape gets pretty complicated with lots of interactions between all the different genes. But we continue to learn more and more about each of these specific syndromes. And as we learn more, we're gaining more opportunity into new ways to treat patients. So standard of care, I'm sure many of you already know, are managing the symptoms of these disorders. So we've got lots of different treatments for developmental therapies, so speech or occupational or physical therapy, behavioral therapy, maybe if you have autism. We also have some medications that can help with some of those symptoms, so like seizures or behavior problems. But when we really want to think about is how do we treat the source? How do we really get to the bottom of what's causing these different disorders? And how do we maybe intervene there and potentially change the trajectory of that developmental profile in a child? And so there's a number of different ways that we can think about this. And one of the big questions that comes out is, will 1,000 different NDD genes require 1,000 different treatments? We hope not, but maybe. And there's a number of groups that are looking at lots of different ways to tackle these problems. So on the left-hand side of the screen, we've got our gene therapies. My group at UT is really focused on gene transfer therapies, so using viruses as little cargo vehicles to get a healthy copy of a human gene into a cell. But there's also gene editing options, or in this third box over here, we've got our antisense oligonucleotides that can up or down regulate expression of genes. There's also dietary therapies. So a great example of that is phenylketonuria, PKU, which they have to have a special diet to maintain because they can't break down certain proteins or sugars. And then there's high throughput screens where we can actually take drugs that are already on the market and say, do they work in different conditions? So maybe there's a blood pressure medication that also interacts with the receptor that's causing a patient's epilepsy. So there's a lot of different innovative ways that all the different groups across the world are looking at to see, do we have new ways to treat our patients? And how do we get those to patients? So this is a big slide that kind of goes through the life cycle of research. So it can be kind of complicated, but we'll break it down and kind of walk you through all the different pieces and how they interact with each other. So starting with the little blue person, in this case, it's an individual with autism. You come to my clinic, we see what symptoms you have. We kind of go through all the different features of your disease, what different body systems are affected. We can think about doing special imaging techniques, looking at brain MRIs or functional MRIs or diffusion tensor imaging to look at tracts of the brain. We can do physiological evaluations, so electrophys, so things like EEG or evoked potentials to see how the brain is functionally responding to different input. And then we can also work with our colleagues that work with animals. So developing different animal models that try to mimic the human disease in a mouse. So making a mouse have autism, seeing how rats or monkeys react when you delete a particular gene, and start to characterize them just like we're characterizing our patients to make sure that the same symptoms we see in our patients are what they see in the animals. You can also do that with cell lines. So down at the bottom of your screen, we can actually take skin cells. So I don't know if any of you have given a fibroblast sample or a skin biopsy, and those skin cells can actually be turned into neurons or brain cells in a dish. And those can sometimes even be grown up into organoids, where you've got a little bit of a mini brain, so to speak, in a dish. And we can use both of these modalities, our animals as well as our cell lines, to test how these new drugs or gene therapies or antisense oligonucleotides are actually interacting and affecting the different models. Are they safe? Are the mice doing okay? Do they look like they have a healthy body weight? Are they surviving? Do they have seizures? How is their behavior in response to the treatment? And same thing in the dish. And then ultimately, we're using all of that information to feed back and say, do we think this therapy is ready to go into humans? And once we're at that point where we say we're pretty confident in our preclinical data, that mouse data or animal data and the cell line data, then we approach in the US, we're approaching the FDA or the Food and Drug Administration. And they are our governing body in the US who are guiding and making sure that patients are going to be safe and passing recommendations on how new drugs get into patients. And there are phases that we go through. And pause right there since I think I right-clicked on accident. So there are phases of clinical trials, and these are pretty standardized, but there's a couple of things that may be a little bit different when we're talking about rare diseases, especially pediatric diseases. So on the left-hand side, you'll see preclinical is always where we start. So thinking back to that last graph, we've got our animals and we've got our cell lines. That's our preclinical studies. Anything that's done in a lab that doesn't involve humans. It may involve human tissue, like our cell lines, but it doesn't involve human patients. Once we've hit the bar of saying, we think things look safe, we think things may be effective, then we can move into our phase 1 and 2 studies. So classically, you'll hear a phase 1 study is going to be a small study done in healthy patients to make sure that the drug is safe and tolerable, and to see how the body interacts with the drug. What's the timing and the half-life? How high does the dose go after it's administered? How long does it take to get into the bloodstream? How long does it stay in the bloodstream? All these kinds of studies to see how the body and the drug are interacting. Phase 2 studies are typically the first time that you're going to go into human patients with the disease of interest. They're usually a little bit bigger study. Now, the caveat here is that in rare disease, we're often combining our phase 1 and our phase 2 studies into a single study. So we'll often go into our disease population of interest with the very first trial with a new drug for a rare disease. They're still very small studies, and the primary goal, or we'll also call it the primary outcome of these studies, is safety and tolerability. We want to make sure that the drug is not harming patients and that they're not having side effects that are so bad that you may not be able to take that drug. And so there's lots of extra testing to make sure that the liver's okay, the kidneys are happy, lots of extra visits to make sure that the patients look healthy and are comfortable with the drug and not having too many side effects. Because these are rare diseases, we're also often looking at efficacy in this first round of testing. So even in your phase 1 and 2 studies, the primary outcome is going to be safety and tolerability, but we'll still be looking at efficacy measures. So our secondary goal or our secondary outcome measure are often going to be efficacy measures. Is the drug doing what we want it to do? Are patients not having as many seizures? Are they hitting higher developmental achievements than we would have expected? Are they talking more? Is their behavior more manageable? All these different pieces that are clinically impacting the patient. Once you've passed through the phase 1-2 studies, then you go into phase 3 and finally phase 4. Our phase 3 study is essentially a bigger version of your phase 1-2 with a little bit more emphasis on the efficacy measures, but you're still always going to be monitoring safety and tolerability. If you hit success with your phase 3, that's typically when you can go back to the FDA as a company and say, we would like to file and say that this drug helps this indication and we want to get it out to patients. And so that's where drugs could be marketed. And then you move into your phase 4 studies, which are these long-term outcome studies, managing and monitoring the safety and efficacy of the drugs over a longer period of time in essentially the general population for that particular drug of interest. So you march through these different phases. There are always exceptions to the rule, but in general, this is how we kind of work with the FDA and move through stepwise to make sure that things are safe and helpful. And we're constantly working with the FDA through every step of this process. Now, going through that in a little bit of a different way, we have these nice bulletins that are produced by the FDA. And they kind of step through this, and happy to have the slides shared so that everybody has this content at home as well to look through closer. But it just goes into a little bit more detail of what I gave you an overview of on the last slide. So we start with our drug development on the left-hand side of the screen, going into the animal testing. Step 2 is talking about our IND application. That's an investigational new drug application. So that's what your investigators or the industry sponsor is going to be filing to convince the FDA that we believe this is a safe enough drug to try in humans, and we think we're going to help patients. Step 3, we go into our phase 1 or 2 studies. Step 5, we go into our phase 3 studies. And then going from 3 to 4, we're moving into drug labeling. And there's a whole bunch of steps that go into that to make sure that everything is accurate and informative and correct. And then you finally get into your phase 4 study where we've got the long-term evaluation of safety and efficacy in a bigger group of patients. That was a whirlwind tour through how drugs make it through the FDA. and get onto your shelf and into the prescription pads of your treating physicians. And so now we can think a little bit about how do we actually design those clinical trials? What's actually happening within the trial itself, and how do we prove that a drug is safe and effective? So the first thing that I want to just remind everybody is rare disease research is messy. We've got a lot of different therapies out there. Most of them are off-label because most rare diseases don't have a specific indication for their disease. And most of the literature out there is going to be in the form of case reports, which are not quite as standardized as some, a rigorous prospective study that, takes a group of patients and follows them in exactly the same manner. We also don't have great guidelines to make sure that everybody's using the same techniques and therapies to the same degree. So before you can say a drug is working, we need to be able to say what the patient would have looked like had we not intervened. So the FDA is always going to ask us, how do you know that that wouldn't have happened, whether with or without your intervention? And that's where your natural history studies come in. So these are observational studies that will follow patients over time and really lay the groundwork for how patients evolve and develop and progress through the different stages of their disease over the lifetime. You really have to know normal before you can know abnormal, or before you can know that you have made a difference or a change. The other caveat to that is that kids are always developing. So in most cases, they will continue to gain new skills over time, even if it's at a slower rate than their age-matched peers who don't have that disability. And so we have to factor that in of like, well, how much change would we have expected? And is this more? How do you also control the environment? So I mentioned we don't have good guidelines. We are using off-label therapies, and patients are all over the world getting all kinds of different therapies and different levels of care and different standards of care. So we always have to think about, how are we going to control for those variables? How are we going to control for all those little differences when we're looking at patients in a clinical trial? And how do you balance the risks and benefits and timing of an intervention? So is there a critical window in which we know we need to intervene when a patient is, 6 months old, or we have to intervene before puberty? Or is there a bigger window, and any intervention at any time point is going to be helpful? And then how long do we need to follow patients to make sure that we've captured that incremental improvement? Is it longer? Is it shorter? And picking our endpoints to help us make sure that we're hitting it at the right time points. And then how do we control for the variables? What are we comparing our patients within the clinical trial to? So in parallel controls, which is more the classic way of doing it, you recruit patients to the study, you put them into two different groups. One group gets treated, the other group does not. And you follow them over time and see how they're doing. And your group that doesn't get treated is compared to your group that does get treated. That's one way of doing it. The other way that's commonly employed in rare disease is to use a historical control data set. So you can take a group of patients that you know what they looked like over a period of time, and then recruit either from within that patient group or without, or outside of it. And you can say, we're going to treat these patients now and refer back to that old historical control group and say, these patients are doing better than those patients are. And so there's two different ways of doing it. There's pros and cons to both. And we'll kind of touch on them a little bit as we go through the talks and more. So our gold standard clinical trial design is going to be your placebo-controlled, double-blinded, randomized clinical trial. So these are great studies when you have tons and tons of patients. They are placebo-controlled, meaning you've got a sham intervention there where some patients are getting the actual investigative intervention, experimental treatment. Other patients are getting a sham treatment or a sugar pill, for example. Double-blinded means that the patients don't know which group they're in and the investigator doesn't know which group they're in. So you can't introduce any bias and say, well, I know you're getting the treatment, so I think you're doing a little bit better. And then you're randomized. So that means that nobody gets to decide. It's at random that patients are put into whichever group, and we're not cherry picking to make sure that we're putting the right patients in each different group. You follow everybody over time, you unblind at the end, and you look at your results to see who did better, our placebo patients or our intervention patients. These are costly, they take a long time. Patients don't really like them because you've got a 50-50 shot of getting treated. And if you're going into a risky situation and time is important, then they're not as favorable and not usually liked as much as patients for patients. Different design techniques have then come in for rare disease especially. So we've got lots of different types of clinical trial designs that we can think about, and I'm going to highlight a couple here. So one is our N of 1 clinical trial design. And this is relatively similar to how we would design a crossover study, but these are actually fairly common now. So just wanted to make sure I touched on them. Essentially, you start with your study subject, and they are running through a period of baseline measures. So we get a nice idea of what the patient looks like now. And then you give the treatment, and then you watch and you take some more measurements. You do some more cognitive testing, communication testing, motor testing. And then you take them off the drug and you watch for a period of time. We'll call that the washout period. And then you reassess and do your measures again. Then you treat again and you reassess, and then you stop it and you reassess. And with each time point, you want to see that you're having an effective change, and then it goes back down, or you have an effective change and it goes back down. And that's how our N of 1 clinical trials are typically designed. Now, with things like gene therapy, they are a one-shot deal at this moment. So for gene transfer therapy, you're getting dosed once and we can't reverse that. So you can't have a washout period with those particular trial designs. So you're getting a nice, good baseline run-in, you're giving a treatment, and then you're following them for a while. So there's some caveats depending on what treatment we're looking at. And then the randomized placebo phase trial is a nice compromise on our gold standard. So for this one, everybody gets enrolled into the trial roughly at around the same time. You're following everybody for those baseline measurements, periodically, MRIs, blood work, cognitive testing, and so forth. And then the randomization comes in as to when you're getting treated. Everybody gets treated, but at different time points. So you can have your assessors, so those, physical therapists doing the motor assessments, your psychologist doing your cognitive battery. They can be blinded to whether a patient's treated or not, and patients are just coming through and getting their testing done. And because they don't know when they got tested, it removes a little bit of that bias of, assuming that patients are doing better or bumping their scores a little bit or being more generous because they are on treatment. You can use a smaller number of patients with these. Patients tend to like them a little bit better because everybody gets treated. They just take a little bit longer to do, and they take a little bit more logistical planning to make sure that they're done correctly. And then finally, we've got our observational studies. And so these are really important to set us up for success on those clinical trials. So we won't know what to do unless we've identified and established what we know the disease looks like. There are several different ways that we can design these studies. Some of them could include disease registries, retrospective natural history studies, where you take, medical charts and review them, or the literature and review them. Prospective studies, where you take a group of patients and you follow them over time in a standardized way. And those kind of link in with our biomarker studies, where maybe you're doing EEGs on everybody to see what they look like, or MRIs or blood work on everybody. So I'm going to show you a couple of examples there. And this is an example that we're working to get published right now with a group of collaborators, where I was working with a group that conducted a retrospective study on a disorder called SLC6A1 related disorder, where we took all the published cases that we could find from all the different databases and tried to plot out what are the different symptoms and how many patients within the group had those symptoms. It's also part of Simon Searchlight. And so we took those patients and said, what did they look like? And then we compared the two. So it's a nice example of how you can use your retrospective data as well as your patient registry data together to try to strengthen your argument that these are the core symptoms of a disorder. And then another great example of a prospective natural history study that also was used as a historical control in a clinical trial for precision therapy is the story of spinal muscular atrophy. So on the left-hand side of your screen is kind of the money shot from their natural history study that was a prospective study, followed patients over time, how many and when did they get intubated, how many passed away of their disease, there's also data in there of how many sat in at what age, how many walked in at what age. And then that was used in the clinical trial for the gene transfer therapy as a comparator arm. And what's really striking is that blue line going across the top of that graph are all the patients that were treated in the clinical trial. And that red line signifies every time somebody in the natural history study had one of their outcome measures, whether it be death or mechanical ventilation. And so you can see the event-free survival decreased in the natural history study and was 100% in our gene therapy. And that was a really powerful way to use a historical control data set to prove that your clinical trial drug is working. So how do we select our endpoints? Endpoints come in a couple different flavors. So we have our biomarkers, which are more of the blood testing, the electrical measurements, so your EEGs, your ERPs, and your imaging data. And that gets paired with our clinical outcome measures, which are more of the functional measures of how a patient is doing. So our cognitive testing, our quality of life scales, our motor battery assessments. And we use a combination of these biomarkers and clinical outcome measures to design our clinical trials to prove that different drugs are working. And you want to make sure that you have a good assessment and handle on how these work in the population by doing your observational studies. So in conclusion, I went through a whirlwind of how you design these different clinical trial programs, but essentially it's marching through using the data that you have at hand. So our retrospective data is usually the most accessible. So looking at case reports that have been published, looking at the patient's charts that you have, and trying to get a lay of the land of what do patients look like. Design a good prospective study to pick those biomarkers and clinical outcome measures that you think are going to be important in the clinical trial, and then use that information to design a really successful and efficient clinical trial where you've got appropriate clinical outcome measures and disease relevant biomarkers to prove the efficacy of this new drug. And ultimately, it's all coming back to the patients, right? So we have our natural history studies. It's a huge burden for you guys. We understand. We have the patient registries. We know that they're asking a lot of different questions and a lot of the same questions often. And then we know that it takes a lot to participate in a clinical trial, but the ultimate goal is that we get better patient care and better treatments for the kids. Finally, building communities is so important. We want to give families a specific diagnosis because it can be really powerful. So it helps you mobilize. It helps you get the scientific community involved and interested in your group. It helps the community become more familiar with your particular disorder by amplifying stories from families that are of the similar vein. And it gives you all a collective voice and a hope for the future that can continue to push treatments forward. So with that, I thank you so much for your time. I thank you for paying attention today. We went through a ton of information. I'm happy to take questions. And I just want to thank again a team of mentors and a team of colleagues at UT Southwestern who are all helping me along the way to make sure that we're doing as much as we can to help our patients. With that, I'll thank you so much and I'm happy to take questions.